Welcome to the Yitzhak Modai annual lecture on technology and economics. This year's distinguished guest lecturer, Professor Ruth Arnon, an eminent scientist and president of the Israeli Academy of Sciences and Humanities. The Academy is a premier body of Israel's scientific community. It numbers 100 members, top Israeli scientists, whose aim is to encourage and to maintain excellence in scientific endeavors in the country. Professor Arnon is the 9th head of the Academy. She was elected president by the Academy's General Assembly in June 2010. Professor Arnon is an advisor to the government of Israel. As a scientist and researcher, Professor Arnon of the Weizmann Institute of Science is a world-renowned immunologist. Her advanced research has been crucial in many developments in vaccines, in cancer treatment, and in parasitic diseases. She was one of the 3 members of the team which develops the first Israeli drug for the treatment of multiple sclerosis, Copaxone, which is marketed throughout the world. Professor Arnon has held many top positions with the Weizmann Institute, including vice president of the institute. She has been the recipient of many varied prizes and honors throughout her life. Among them, the Robert Koch Prize, which honors microbiologists who make groundbreaking discoveries, the French Republic's Knight of the Legion of Honor, the Hadassah World Organization Exceptional Women Award, the Israel Prize, the Wolf Prize for Medicine, and the Rothschild Prize in Biology. In 2009, she was elected a member of the prestigious American Philosophical Society, comprised of top international scholars from a wide variety of academic disciplines. We are honored that she will be our speaker today. It is my pleasure now to call upon Ruth Arnon to deliver her speech.
Good morning, everybody. I'm very pleased to be here. The Technion is not new to me, and I'm very pleased and honored to be here. My husband is a graduate of the Technion. My brother was a graduate of the Technion. My nephew and my niece are graduates of the Technion, and even the grandson of my brother. So I have strong relations, and I have been for several years a member of the Curatorium of the Technion, so I really feel at home here. I have another remark to make. As the president of the Israel Academy of Sciences, yesterday we had our annual meeting for election of new members, and I'm pleased to announce that Professor Moti Segev of the Technion was elected member of the Israel Academy of Sciences. So congratulations, Moti Segev. The topic that I chose to speak about today is one that is close to my heart, and it deals with vaccine research and new development and the challenges and prospects that are before us. I chose to begin this lecture with this picture, this photograph. I don't know if you recognize this place. This is the cathedral in Siena, in Italy. Beautiful cathedral. Now, what I want to point is this strong structure that you see here. You see that it is a wall, like a gate, like huge wall that containing a door. You can see the size of the wall. This picture is taken, of course, now. But in the 14th century, Siena was one of the richest cities in the world. It had many merchants and commerce in it. And the cathedral was built before in the 12th century. And it became too small for the population, and they decided to enlarge it. They decided to enlarge it by building another part, the apse that will go here, and this was supposed to be only the nave of the new cathedral. So they started building. On the floor, you can see even the bases of huge pillars, And then came the plague. The plague in the 14th century. Approximately more than a third and close to half of the population of Siena have died during the plague. At the end of the plague, there were not, first of all, it was not needed anymore to enlarge the cathedral. There were no builders, there were no artists, there were no people that could have, and they have abandoned the building, and they have decided to keep this wall in memory of the plague of the 14th century. So this is just in order to demonstrate the power of diseases that happened during the past. And diseases were not a few, there were quite many. One of the worst killer was smallpox, אבעבועות שחורות. And this was from the ancient history. We see here mummies that were taken from Egyptian graves with signs of smallpox. And on the painting, on the wall paintings on caves, you can see also patients with smallpox. It was a huge killer that killed every year millions and millions of people. And then, about a little over 200 years ago, a country doctor in England made an observation. He saw that milkmaids had very beautiful faces. They had no scars on the faces that usually were remnant on the faces of people who had smallpox and then survived. And he asked himself, is there any connection between the fact that they are milkmaids and the fact that they do not have smallpox? And he observed that the cows that were in the village had a disease that was similar to smallpox, but it was a disease of cows that did not cause too much damage to the cows. It's a much milder disease. So he took some materials from the cows and made an experiment, a clinical trial. He injected a little boy with this material that he took from the cows, and then this boy was exposed to people who suffered from smallpox, and he did not get ill. And Jenner have tried to publicize his findings and has talked about it with many of his friends who are doctors, and other people started applying this technology, and eventually this is the vaccine that was used for the eradication of smallpox, and now smallpox has been completely eradicated from the world, and there's no smallpox anymore. So this is the power of a vaccine. However, if you look at the history of vaccinations, Jenner's vaccine was a fluke. By chance, cowpox and smallpox viruses are similar, so that one could protect against the other. But there are no other diseases with the same luck, I could say. Only 100 years later, about a little more than 100 years ago, Pasteur discovered that diseases are not caused by dirt, but they are caused by microbes, either bacteria or viruses, and these are the agents that cause the disease. And Pasteur was the first one who developed intentional attenuation of the microbes so that they could not cause the disease anymore, but they still produced a material that when you put this material into people and into animals as well, Pasteur was very active also in protecting animals against diseases, and you can protect them by this procedure. And the procedure is called vaccination because of the name vacca. Vacca is the Latin name for cow, in honor of the first vaccine that was developed or invented by Jenner. Today we have many types of vaccines. We have vaccines that are for bacterial diseases that are based on the whole bacteria or on small parts of the bacteria, against viruses, again, based either on the whole virus or the virus subunits or other. And all of them are based on the pathogen that induces the disease, but you treat it in some way, either kill it or attenuate it, so that by injecting it to people, this material does not cause the disease anymore, but protects against further exposure to the microbe or bacteria or virus that cause the disease. In addition, there is another type of vaccine that I will come to it later, which are called passive vaccines. I mention it here, but I'll talk about it later. Now, the development of vaccines, we all know, has been the most successful and cost-effective contribution to public health. This is known. During the last 2 centuries, vaccine has led either eradication or drastic disease in the incidence and morbidity of many diseases, many of them very severe. And the available vaccine today that we use are mostly prophylactic, namely in order to prevent the disease towards the next exposure. I show you here just the results of the reduction in various infectious diseases. As you see, rheumatic fever, hepatitis, mumps, measles, tuberculosis, all of them, as a result of the development vaccine, the incidence of the disease dropped really drastically. And not only the incidence of the disease itself, for example, in the case of measles, not only the incidence of measles dropped, but also of diseases that are the results of exposure to measles, namely encephalitis, that sometimes is a side effect of measles, or SSPE, which is another disease, mainly among the Arab Jews in Israel, that suffer from a post-effect of measles, and this also dropped considerably, drastically, due to the use of the measles vaccine. And of course, these are not the only reason, but there are many others. But because of this, or due to this, life expectancy rose really dramatically. If we look at the life expectancy in classical Greece and Rome, you won't believe it. It was 28 years. This was the average life expectancy during the time. And even when we reached the early 20th century, the average life expectancy was only 35 to 40. Of course, there were some old people, but this is because there were many babies and young children that died, and the average was approximately 35 or 40. Currently, the average world is 66% or close to 67%, and in many of the developed countries, it's 80 and even over 80. In Canada, Australia, France, Japan, and Sweden. So this is, of course, not only to vaccines, because also hygiene conditions have changed. We have more clean water. There are also antibiotics that affect life expectancy. But vaccines is one of the common practices. Vaccination is the practice that really prevents childhood diseases, and most of the vaccines that we have are used for children, for pediatric uses, and they really save children for many of the diseases that we used to have, and now we don't hear about them almost anymore. This is a list of the diseases against which we have vaccines today. I will not read the list, but as you can see, it's a huge list, which these diseases used to be. When I was a child, we used to have diphtheria. From time to time, there was a breakout of cholera, other diseases like hepatitis, measles, mumps, all of this. And today, these diseases practically do not exist anymore in the developed countries. So if everything is so good, why do we need to do research on vaccines? And this is because there are still shortcomings of the present vaccines. One shortcoming is that conventional vaccines are based on the pathogen causing the disease, and some pathogens cannot be grown in culture in order to produce the vaccines. Not all the pathogens are amenable to attenuation, so suitable vaccines cannot always be prepared. And in some cases, there is a biohazard in preparation of a vaccine of insufficient attenuation. There is a known case in Egypt where a bad batch of a polio vaccine that was not sufficiently attenuated was used, and a whole group of children that was vaccinated, all of them got polio. And in addition, a phenomenon that exists today is the genetic variation viruses that leads to new strains of the virus, and thereby to inefficacy of the vaccine that was prepared against the previous strain that we knew. And this is known in influenza. You know, every year there are new strains of influenza. The old vaccine is not effective anymore, so a new vaccine has to be prepared every year in order to protect against influenza. And I will come to it again later. There are also at least alleged safety concerns about influenza. One of them is the risk of allergy and anaphylaxis. For example, all the vaccines against influenza are prepared, the virus is grown in eggs. To people who are allergic to egg proteins, this can be unpleasant. There was a rumor that there is a link between the measles, mumps, and rubella vaccine and autism. This was proved wrong, but there are some people who are still afraid of vaccination because of these alleged risks. Even later in life, there are some people who complain about allergy that is due to vaccination. This also, in most cases, was proved wrong, but still people do believe in it, and in many cases it prevents them from vaccination. And don't get it wrong. If vaccination will stop, the diseases will come back. We know there was a case in Great Britain. They decided to stop vaccination against whooping cough, and within 1 year, the disease came back, and they just realized that they have to continue to vaccinate the children. There is also a risk of autoimmunity because some vaccines contain components that are similar to our own body components, and they can lead to autoimmunity, and again, I'll come to it later. Most of the vaccines that we know are prophylactic. Because the infectious diseases that we know, most of them are of short duration. So you must vaccinate and protect the person before the encounter with the disease-causing agent. Otherwise, it is too late in most cases. So because of this, most widely use of vaccination is for pediatric use, namely for children. In the first 1 or 2 years of life, children are vaccinated with practically all the available vaccines, and most of them give protection for a very long time, many of them even for life. But the challenge that we have today is to meet future demands, not only the short-term infections, but in many cases emerging and re-emerging pathogens that cause new diseases, and I will give a few examples in the next slide. The increased incidence of autoimmune diseases that we must find solution to, and can we use vaccination for this? Fighting cancer. Can we immunize, can we vaccinate people against cancer? Because of all this, there is also a new idea, a new approach, and this is the development of therapeutic vaccines that will be used not as a prophylactic agent in order to prevent a disease, but to treat after the disease has already occurred, and this will be the second part of my talk. Emerging and re-emerging diseases, there are many examples. The first one is the Black Death, which was in the 14th century. There was the influenza pandemics in 1918 that killed 30 million people only in Europe. More people than were killed in the entire World War I were killed in 1 year because of the influenza. We all know about the AIDS that existed at the present. Tuberculosis, there's re-emergence of tuberculosis and even resistant strains of tuberculosis that are resistant to antibiotics. You remember SARS several years ago, and one of the big fears is bioterrorism, the use of viruses or disease-causing agents as a biological warfare. And for all this, can vaccine help? There are many factors that are involved in the emergence of diseases, and I will not go through this list, but there are microbial adaptation and change, human susceptibility to infection that changes, climate and weather, and poverty, war, and famine. All of these are factors that can affect the incidence of emerging and re-emerging diseases. And there are also additional aggravating factors. Today, there is no distant disease. Any infectious disease outbreak, wherever it originates, has the potential to influence the entire world and within a very short time. People use airplanes. It is known about SARS and also about influenza. In 1 or 2 days, the disease goes from East Asia to Canada or to the United States. There are also dangerous interactions in the ecology between people and animals. SARS was spread by rodents. Avian flu is transmitted by birds. And also, AIDS came from monkeys because of the destruction of forests. So the apes or monkeys that used to be in the forest, they have a disease that is similar to AIDS, but when the monkeys reached closer to human habitation, they infected people, and this led to AIDS. So what are the countermeasures that are being taken? First of all, there are global actions. Early warning of infectious disease, establishment of new international laws for the management of global health issues, establishment of global infrastructure for vaccination. This is very important in many countries, but also scientific efforts in order to develop more effective vaccines. And a case in point is influenza. Influenza, as I said, is an old foe that raises his head from time to time, and there is a big fear for a new pandemic that may occur either now or years to come, and there is a lot of preparedness that is needed for this. The most recent one is the H1N1 swine flu next year that still circulates in the population. And these events call for the development of an effective prophylactic vaccine that will be universal. By universal, I mean that it will be effective against many strains. Now, for this we have to, this is a little bit of a science, just on the tip of the fork. This is the structure of influenza virus. And as you see, on the surface of the virus, there are two major proteins. One is here is red, which is the H, and the green one is the N. And these two proteins change every year, and sometimes even more frequently than every year. And because they are on the surface of the vaccine, and the immune response that is raised by the vaccine or by the virus, first of all, the antibodies see these proteins that are on the surface of the virus, and if there is a change in the virus, they don't recognize it anymore, and they are not effective against it. So what is done today, and all the available vaccines are based on it, the WHO, the World Health Organization, every year before the season, which means sometimes during the summer, they monitor the different strains of the virus, because the virus usually comes from the east to the west, from East Asia through Europe to the Western Hemisphere. So they monitor the different strains of the virus, and they decide according to that on 3 different viruses, and they tell all the companies, you in the vaccine for next year, you use virus this one and this one and this one. And all the companies start growing the virus and preparing the vaccine from it. And the vaccines that we have today are either the whole virus that is inactivated, or you split the virus and you use it as a vaccine, or you use just a complex of the 2 proteins. This is called the subunit vaccine. Just a complex of the 2 proteins is isolated and is used for vaccination. And recently there is also a live vaccine, attenuated virus that is being used. But all of them are based on the total proteins that are used for vaccinations, and when the protein change, the vaccine will not be effective anymore. A new vaccine will be needed. The approach that we took in our laboratory is a new concept. We use small regions of the viral proteins, not only these 2 proteins, but also additional from the interior of the virus. We express them by genetic engineering and use a mixture of this small region for immunization. And when we immunize mice against it, as you can see in the upper, if you immunize all the mice that we immunized with the mixture of 3 components like this survived, whereas all the control died, and a mixture of only 2 was not enough. We needed a mixture of the 3. And protection lasted for a long time, even 7 months after the vaccination. All the vaccinated mice survived when they were exposed to the virus, and all those that were in the control group, they all died. So this is a wonderful vaccine for mice. Can you use it also for people? So we worked on it, and we identified different regions of the virus that should induce protective immunity in people, and indeed a mixture of them was prepared, and we used this mixture for immunizing what we call humanized mice. What are humanized mice? These are mice that by radiation, you destroy their immune system, and then you transplant them with white blood cells from people. And then, so they are mice, but they have the immune system of humans. And these mice, when they were immunized, not only that they were protected against the disease, but they produced human antibodies, and these human antibodies indeed were specific against many strains of the virus. So this could act as a universal vaccine protecting against many diseases. Now, on the basis of this, a small company was established, BeyondVax, and the idea is to produce a universal vaccine. They have preclinical trial, and they are already after phase 2 clinical trials that proved effective. There is still a long way, as I know from my previous experience, but if this vaccine succeeds, it will have competitive advantages because it will be universal against many strains of the virus. And by the way, people that participated in the clinical trial 2 years ago, they tested in their blood, they tested for antibodies against the virus of last year, which did not exist even at the time that they were vaccinated, and they still recognized it and were, I don't know if it were protected, but they still responded against the virus. So this is a promising approach. A vaccine like this could be multi-season with no need to vaccinate every year. It will be safe because it will not have egg proteins, and its production procedure is shorter than the usual vaccine. But we only have to see if phase 3 is successful and if it is working. I would like to move now to another aspect, which I mentioned in the beginning, and this is therapeutic vaccines. Until now, I talked only about prophylactic. You have first to be vaccinated and expect that due to this vaccination, you will be protected against future encounter with the disease-causing agent. But is there a possibility to use vaccines as treatment, as therapy? So first of all, we know from long time ago that there is what is called, I mentioned it in the beginning, a type of vaccine that are called passive vaccines. What are passive vaccines? You can immunize animals and use the antibodies that you isolate from them for therapy. And this was known for example, in cases where the disease is caused not by the bacteria, but by toxin, like diphtheria or tetanus. What causes the disease is the toxin that is secreted by the bacteria. And in this case, antibodies against the toxin were prepared in horses and in other animals, and they were used as a treatment for patients who were already ill. We know for tetanus that this has been used too. And also in the case of snake venoms, there are many anti-sera against different snake venoms. In Israel, we have against the Vipera palestinea. There are stocks of these anti-sera that are prepared in horses. They are available, and if somebody is bitten by the viper, and I know quite a few people that have been saved. They would have died without this vaccine, but they have been saved. And this is a passive vaccine that is used as therapy and not as prophylactic. But there can be also active vaccination that could have a therapeutic potential. For example, for chronic or long-term infections, for example, AIDS or tuberculosis that lasts for a long time, or some parasitic diseases like malaria. In these cases, even conventional vaccines, if they are prepared in an adequate way, might still be effective. And I will give you some example only in the case of vaccine of HIV, because in HIV this is still a disease that needs desperately a solution. There is no point in vaccinating the entire population in the world against HIV. Even in the regions of risk, there is no point in vaccinating the entire population. So a therapeutic vaccine has to be found. And we have some vaccines that are based on proteins of the HIV. Theoretically, even using small regions of the proteins can be used. And there are several vaccines like this that are in development. Some that passed already phase 2 trials. Phase 3 trials in large number of patients is still needed, but they are not available yet. On the other hand, there are therapeutic vaccines against other diseases that are already in use. For example, there is a vaccine against tuberculosis, a vaccine that can be given in a patient that already suffers from the disease, and a company, Archibald, is producing this vaccine, and they are now in phase 3 trials. It is effective, and probably it will be used. And same thing about malaria. Some vaccines have already been prepared and are in clinical trials and look very promising. Another case in which therapeutic vaccines could be effective is in the case of cancer. In this case, again, prophylactic by vaccination is impractical. So in practice, one has to focus on therapy, either use drugs or, if it is possible, to prepare vaccines that can be used. So vaccines today are being, again, there is a very intensive research on this, using various components of the cancer cells that are specific for the cancer cells and by which they are distinguished from normal cells, and they are used for preparation of vaccines. They are being used against several diseases, and none of them is approved for use, but a strong effort is invested in this direction. But what is already working is what we call passive therapeutic vaccines. These are antibodies that are prepared against particular component of the cancer cells that are specific for the cancer cells. And these are what I call passive vaccines, because these are antibodies that are prepared in advance. In this case, using new technology, which is called monoclonal antibodies. And there are quite a few antibodies like that are already available as drugs. Rituximab, that is used very widely against B lymphomas. Herceptin, that is used against metastatic breast cancer. Maptera, that is used against chronic lymphatic leukemia. And when I say used and very successfully used. Erbitux, that is used mostly against metastatic colonic cancer, and Avastin. I'm sure that you are familiar with these names. Every year there is a battle about the basket of drugs, of therapies, whether to include more and more, because these monoclonal antibodies are quite expensive, and in some cases they save lives. Herceptin really save lives. I know many women that had metastatic breast cancer, were treated by Herceptin, and they are completely tumor-free for years and years. So it really is helpful. And some are only prolonging life, but still they are effective, and it's one of the best examples of a passive vaccine that is also a drug. Another example where therapeutic vaccines can be used is for the treatment of autoimmune diseases. Now, what are autoimmune diseases? These are disorders when our own immune systems that usually is geared in order to identify anything that is foreign and to remove it. And by this, we fight against infections. But in some cases, something gets wrong, and our immune system recognizes our own body components as if they were foreign and respond against them. And this causes several diseases. And in this case, therapeutic vaccines could also be appropriate and an option. And another option could, of course, be monoclonal antibodies that will be specific, and both of these options have been taken. And again, a case in point is multiple sclerosis. This is caused by autoimmune response of our body against the myelin. Myelin sheath is the envelope that covers have the insulation of all the nerves in our central nervous system. And when there is a damage to the myelin, like in an electrical cord, when there is a damage to the plastic, eventually there will be a short circuit. So the same thing occurs in multiple sclerosis. There is damage to the myelin, and eventually there is neurological damage. Now, the drugs that are available today all actually try to reduce or to modulate the immune response. There are 3 beta interferons that are used, and the Copaxone that we have developed that is also one of its mode of actions is by modulating the immune system. Another drug that is being used today is Tisabri, which is a monoclonal antibodies against a cellular adhesion molecule, and this also is helpful in modulating the immune response and in preventing the damage of the disease. I want to say just a few things about Copaxone and its development. It is a synthetic polymer of the amino acid, and we came about it because we designed it in order to simulate the protein in myelin that is supposed to be involved in the induction of the disease. And indeed, it resembles it, but by itself, it cannot cause the disease at all, and it is not an immunosuppressor. But when we tested it in various species of animals, we could see, although it cannot induce the disease, but it can reduce the incidence of the disease from 80% or even 100% in rhesus monkeys and in baboon to approximately 20%. And according to that, we collaborated with several neurologists, one of them in the United States. He performed a phase 2 trial, and as you see, there is a huge difference in the incidence of exacerbation between the control group in red and the treated group with the Copaxone. And according to that, eventually the FDA approved it as a drug, and now it is marketed worldwide. About 250,000 people in the world use it, and it is marketed for approximately $3 billion per year. So this is quite successful. But what is more important to me is the effect on patients. This is a follow-up on a large number of patients, and you can see the incidence of exacerbations of this disease, which comes in relapses and remission, drops down by a factor of 10 from more than 1 attack every year to 0.12, which means on the average 1 exacerbation per 8 years. And this allows patients to live. I know patients that are treated by Copaxone for 25 years and a little more, and they are still practically free of exacerbation and can lead normal life. And this is a huge difference in the life of multiple sclerosis patients. I knew these patients before drugs were available. So this is the real satisfaction for a scientist like me. I just want to mention that today there are therapeutic vaccines on the process of development to several other autoimmune diseases, diabetes type 1, which is juvenile diabetes, rheumatoid arthritis, myasthenia gravis, and lupus. These are a few diseases for which such therapeutic vaccines are being developed. And we should still keep in mind therapeutic vaccines, which means the monoclonal antibodies. I give here a list of monoclonal antibodies. You might have heard about them: Humira, Remicade, Enbrel, Cimzia. These are all monoclonal antibodies against a certain protein, TNF-alpha it's called, and they are in use today. This is really the therapy. Autoimmune diseases such as rheumatoid arthritis, psoriasis, Crohn's disease, and others, and several others are on the verge of being developed. So what are the conclusions? First of all, prophylactic vaccines have been with us for many years, and they have been instrumental in eliminating the major killer diseases. More efficient vaccines are still under development. Therapeutic vaccines is a challenge, but they constitute an attainable goal for future research. The potential applications are several. First of all, to arrest chronic infections, treatment and therapy of cancer, treatment of autoimmune diseases, and thereby they have a tremendous potential for benefit for public health in usual. So thank you very much for your attention.
this concludes the Modai lecture. Thank you very much.
Do